Assalamu alaikum. Welcome back to lecture 2 of DNA and design. In this lecture, I would actually like to begin doing some real science now, but I'd like to actually begin with a detour, which I know is something unusual, but I would like to talk about a more macro issue before we get into DNA, its function, its chemistry, and so on. Perhaps start with something a little bit more familiar that illustrates the paradigm that I would like to bring across in these lectures, which is that advancing scientific knowledge is pointing to the notion of design rather than notions of randomness, chance, and unguided evolution. And this has to do with a controversy regarding something very familiar to all of us, the eye. And this controversy, as we have learned more, has swung the pendulum back, at least in my opinion, toward notions of design. So let us begin. So what is this particular issue regarding the eye? Well, I'd like to begin with Sir Isaac Newton. And Isaac Newton, as you know, is considered probably the greatest or one of the two greatest physicists and scientists who have ever lived. The other name that would come up, of course, is Albert Einstein. And during Newton's day, and for Newton personally, looking at both natural phenomena like the orbit of the planets and looking at human physiology was clear evidence of God's creative activity, was clear evidence of the supreme intellect behind this world. And so in his general scholium, Newton says of the solar system, this most beautiful system of the sun, planets, and comets could only proceed from the counsel and dominion of an intelligent and powerful being. And of course, he was talking about God. Now, in the realm of human physiology, Newton, as you know, did a lot of work on light and optics as well as physics, and was particularly fascinated with the human eye. And here you see just a rough diagram of the structure of the eye, that light comes in through a lens and through the pupil and is reflected back on the retina, and then that sets off electrical activity in the nerve cells in the retina, which travel from the back of the eyeball, from the retina, through the optic nerve, and eventually ending in the visual areas of the brain in the occipital cortex, more specifically something called the calcarine cortex. But in any case, during Newton's day, the rough anatomy of the eye was known, and Newton found the eye to be a marvel. And again, one of the good pieces of evidence for divine design. And so he says in a draft of the hypothesis concerning light and colors that one principle in philosophy is the being of a God or spirit, infinite, eternal, omniscient, omnipotent, and the best argument for such a being is the frame of nature, and chiefly the contrivance of the bodies of living creatures. So here is the strongest possible statement of divine design. And then talking specifically about the eye, he says, certainly he that framed the eyes of all creatures understood the nature of light and vision. He that framed their ears understood the nature of sounds and hearing. So this was the general outlook of science in Newton's day, in the 1600s, in the 1700s, and so forth. That outlook began to change in the 18th and 19th centuries, and as we have already said, under the science of Darwin, under the philosophy of philosophers such as Hume, the pendulum swung away such that scientists began to now reject the idea of design. Okay, so now let's get to the specific issue at hand. In the diagram on your left, you see a blow-up of a portion of the retina, which is that part in the back of the eye, which receives the light and sends the nerve impulses back to the brain. And as we began to learn more about the structure of the eye, especially with the development of electron microscopy, something very interesting was revealed. And if you take a look at the diagram on the left, you will see an enlarged illustration of the retina, and you see things labeled rods and cones, and they are the photoreceptors of the eye. Now there's so much to say about the eye, what the rods do, what the cones do, but let's ignore that for now. What was interesting is that the rods and the cones, which react to the light, then project their axons to other cell bodies forward. And those are the blue cell bodies, what are labeled as number 3 in the diagram. Those cell bodies then coalesce into nerve fibers and nerve impulses that collect into the optic nerve, which is labeled number 4. And if you now look at the diagram on the right, you see the arrow that says path of incoming light, and that light travels through these cell bodies, the connecting nerve cell layer, the ganglion cell bodies, to get to the rods and cones. They react to the light, then they send their impulses forward again to those secondary neurons, the connecting nerve cell layer, the ganglion cell bodies, and those fibers then go to the optic nerve. So why is this setup interesting? It is interesting because it means that the light has to travel through a bunch of cells and nerve fibers first to get to the rods and cones. So whether you look at the diagram on your left or the diagram on your right, you see that the light is not first hitting the rods and cones and then going to other cell bodies, but it has to travel through a mesh of cell bodies to get to the photoreceptors. And this is called the inverted retina. And in this diagram, we've tried to make it a little bit more explicit. In the verted retina, which is found in some lower animals, the light first hits the photoreceptors, then they coalesce into nerve fibers. In the inverted retina, which we humans have, you notice that the light has to go through nerve fibers and other cells first before it gets to the rods and the cones. And this was felt to be an engineering design flaw. Why would you make the light have to go through a bunch of cell bodies and nerve fibers, blurring it and scattering it before it gets to the actual photoreceptors of the eye? And so when this detailed structure was discovered, this was taken by the anti-design camp as very strong evidence that there is no such thing as divine design. And Richard Dawkins, again, one of the strongest proponents of atheism, a leader in the so-called New Atheist movement, in his famous book, The Blind Watchmaker, in the late 1990s, used this to sort of mock the notion that the eye was a design marvel showing evidence of God. And I gave you here a quote from the book. Any engineer would naturally assume that the photocells would point toward the light with their wires leading backwards toward the brain. He would laugh at any suggestion that the photocells might point away from the light with their wires departing on the side nearest the light. Yet this is exactly what happens in all vertebrate retinas. And so this was sort of a smoking gun in the hand of the evolutionist camp. And I am not, by the way, against evolution. I just believe that it is only part of the story. I believe in guided evolution, but that is another topic. But in the hand of those who believe in entirely pure chance, randomness, and so forth, these sorts of design flaws are very strong evidence that yes, things did occur by chance because chance does not lead to perfection. It just leads to something functional. And so when you do an internet search, you will find a lot of stuff about the backwards human retina and is it evidence of poor design and are human bodies poorly designed and so on and so forth. And again, I think we've explained the issue enough that we all now feel comfortable what the issue is. And again, in the hands of people like Richard Dawkins, this was taken as clear evidence against intelligent design. And by intelligent design here, I mean design by the divine intellect, because there was this obvious design flaw in the human eye. Amazingly, about a decade after the mocking statements of Richard Dawkins, new discoveries in the structure of the retina were made. And it was discovered that the nerve cells in front of the rods and cones through which the light has to pass, those cells that were thought to be a design flaw, are known as Mueller glial cells. And they're actually living optical fibers, and they transport the light through the inverted retina. And you can see here a blurb from the Proceedings of the National Academy of Sciences, one of the most prestigious scientific publications in the world, stating that the Mueller cells collect the light at the retinal surface and guide it to photoreceptor cells on the opposite side, and therefore there is no distortion of the light as it passes through this layer of cells. And it turns out that that was just the beginning of the story. Further work has shown that these cells do much more than simply passing the light back to the rods and cones without distortion. Here is a blurb from the organization physics.org. And by the way, I want to make it clear that these are purely scientific publications. Nobody here has a design or theistic agenda. And let's look in some detail at what they're saying. And so they are quoting a paper in Nature Communications. And again, those involved in the scientific world know that the big publications in the world of science are things like Nature, the magazine called Science, the Proceedings of the National Academy of Sciences. So these are discoveries that have been vetted at the highest level and gauged important enough to publish in these first-rate journals. So it says that these highly refractive index cells spread an absorptive canopy across the retinal surface and then shepherd the photons through a low scattering cytoplasm to separate receivers, much like coins through a change sorting machine. And a new paper in Nature Communications describes how these wavelength-dependent waveguides can shuttle green-red light to cones while passing the blue-purples to adjacent rods. So what this blurb from Nature Communications means is stated by the author of the article, John Hewitt of physics.org, that basically having the photoreceptors at the back of the retina is not a design constraint, it's actually a design feature. These cells basically sort the light to where they need to go. Those photons that need to go to the rods are sorted to the rods, and those photons that need to go to the cones are sorted to the cones, rather than simply having the light fall indiscriminately on the rods and cones. And so you see that John Hewitt says this turns out to be a design feature. And I will just read to you what is up on the right there, just so that we can sort of digest it together. The idea that the vertebrate eye, like a traditional front illuminated camera, might have been improved somehow if it had only been able to orient its wiring behind the photoreceptor layer, like a cephalopod, is folly. Indeed, in simply engineered systems like CMOS or CCD image sensors, a back-illuminated design manufactured by slipping the silicone wafer and thinning it so that light hits the photocathode without having to navigate the wiring layer can improve photon capture across a wide wavelength band. So that's what you would do if you were constructing an artificial light capturing system. But real eyes are much more crafty than that. So basically, the bottom line is that as we have learned more science, what was once thought to be a design flaw, evidence for chance, evidence against intelligent design, has turned out instead to be a design feature speaking very strongly for an intelligently designed system. And the reason that I wanted to start our talk about DNA with this example is because I believe it is a perfect paradigm and perfect illustration for what has also happened in the world of DNA. Since the discovery of DNA in the 1950s, the discovery of its structure, moving forward to our current decades, we will inshallah in the coming lectures begin by reviewing a little bit about what DNA is and how it works in the cell, and then take up various aspects of the science connected with DNA that I believe points significantly to intelligent design, and the more we learn, the more strongly it points in that direction. Thank you for listening. Take care and see you inshallah in the next lecture. Assalamu alaikum.